Video Tutorial 2, Data Visualization. In this video we're going to talk about one of the most important feature of tappAS, its visualization. It includes general summary visualization, gene annotation data visualization such as transcript or protein structure and analysis specific data visualization. General summary visualization. What do we mean by general summary visualization? When you create a project in tappAS, the tool plots summary statistics for your data. These are referred to as general summary visualization. For example, you can find out how many genes are coding and which of them produce a single or multiple proteins. The next type of data visualization that tappAS can provide is related to gene annotation data. In this case, we have multiple options. For instance, we can visualize transcript or protein structures aligned, as well as their functional information to understand the differences among isoforms. Also, we have genomic information relative to the gene structure for each transcript. Gene ontology graphs. Graphic charts to see the expression of the gene, its transcripts and proteins. And if you are interested, you can access the real annotation file data for a specific gene. Finally, once you have run an analysis, you may access analysis-specific visualization options, such as gene summary for the differential isoform usage analysis abbreviate to DIU, or the different plots in differential feature inclusion analysis abbreviate to DFI. That said, let's get started. As you can see, we loaded a new case control project named Videotutorial Case Control. We want to remind you that you can create or load your projects by the button Projects. When you open a project, tappAS shows you the same windows that we saw in the last tutorial. We can see our data in the center and the general summary at the bottom. And now, we will show you the many visualization options that tappAS includes. To access the rest of the general summary visualization, we have to click on the plus button, next to the general summary subtab title. A similar plus button is included on the top panel and on the right side of the main table. A new subtab will appear, where we can access a menu including all of the general summary visualization options. As an example, I will show you the genes summary. The genes summary represents how many genes are coding and non-coding, and how many of them have single or multiple transcripts. Furthermore, this option provides a density plot for each experimental group. Depending on the nature of your experiment, this plot could include time points instead of experimental groups. Next, I will show you the annotation summary. This table contains, for each annotatino feature, the number of sub-features indicated by the number of feature IDs, and the number of occurrences of this feature in your data indicated by the number of entries. The source column indicates the database from which we obtained this data. For example, in the case of the Microna database, we have 718 different feature IDs and a total of 162,957 entries in all of our data. If you want to see more information, you can right-click and select Show Source Details. On the right side, you can see a pie chart containing the proportion of information that is drawn from each source. Feel free to click on any other of the options in the General Summary Visualization menu and explore the many features that tappAS can offer. Keep in mind that, in the main panel, we are seeing the transcript information table at all times. If you are interested in accessing the gene or protein table, all you need to do is click the plus button right next to the tab, as we mentioned before. Now, we will move on to gene annotation data visualization. Now, to access a new set of visualization options, I will use the search box to find a gene of interest, the KIF1B gene. To search for your candidate, you can introduce gene, transcript or protein ID in the search box, as well as functional terms associated to them. Once you complete the search, click on the right mouse button. In this new sub-menu, there are 3 different options to click. The first one opens the tappAS visualization tab at the bottom of the screen. The second one allows you to open online references about this gene in its website, either Ensembl or RefSeq. And the last one exports the search result table, or a selected subset, as a file. The export option has different configurations, but we will see that in another video tutorial. We will now click on the Gene Data Visualization option and open a gene-specific tab. By default, the first subtab we can see is the Transcripts Visualization. This screen shows that the gene KIF1B has 4 different transcripts. The transcript structures are aligned by CDS regions for a more intuitive comparison. We represent the CDS region as gray rectangles, and the UTRs with black lines. The rest of the symbols you can observe are functional features. We advise you to use your mouse to explore the structures, since extra windows will appear and show you additional information. For example, the start and end position of a CDS, or the transcript length. Also, tappAS provides splice junction information. And, furthermore, tappAS also shows extra information, such as polyadenylation or microRNA binding sites, as the legend shows. We can customize this visual information in several ways. Click on the first icon on the left-hand side panel to see customization options. As an example, I will focus on the Annotation Features filter. In the emerging window, I will select the information I am interested in. If I wanted to focus on microRNA binding sites, I would clear the entire selection except for the microRNA data from MRWAC, and then select OK. Now, the annotation features shown have been filtered according to my selection. The next kind of gene visualization option provided by tappAS is protein annotation. To access this, we need to click, again, on the plus icon and select the protein option in the menu. The KIF1B gene codes for 3 different proteins. This view is similar to the transcript view. In this case, CDS regions are represented as a black line, while the rest of the symbols represent functional features, such as domains and motifs. On the left-hand side menu, we can access customization options. In addition to filtering, we can sort the information that is shown. To do this, I will select the Sort By option, and select a sorting criteria, for instance, expression level. If we click on it, protein models are sorted by their expression level, which is represented left to each protein. Last, we will see genomic annotation, which shows information about the exons in 5 UTR and 3 UTR. If you keep the mouse on any exon, tappAS shows you its genomic position. You should know that every graphic you find in tappAS can be exported. Click on the right button to access the Export option. In this case, you can also use the left-hand side panel to customize visualization or further explore the annotation. Remember that left side menus vary depending on the selected graphics, and you should check the available options in each window. In the Genomic Annotation window, you can export the image, zoom in, zoom out, or click on the Help button to know what we are seeing. The Gene Ontology tab shows a tree with the different processes where this gene is involved, and their hierarchy. You can switch between 3 Go categories: biological process, molecular function and cellular component, and zoom in by double-clicking, and zoom out by pressing the key Shift while double-clicking. On the Expression Charts tab, you'll have access to the total expression level of this gene, the expression level of each transcript, and the CDS expression. We infer CDS expression by adding the expression of transcripts encoding the same CDS. In the case of the KIF1B gene, the transcripts represented by the blue and yellow lines encode a single CDS, represented by the blue line in the CDS expression plot, as you can see in the emerging window. Note that the red and green transcripts stay the same. The charts represent the mean expression level with the standard error of the mean (SEM) for each group or time point, depending on whether you are working with a case control experiment or a time series. All these plots can be exported as we said before. The charts can also be moved to another window. To do this, select the Float subtab option on the left-hand side menu, and move it back into the main window in a similar way. Last, I will show you the tappAS annotation file for this gene. This table represents the structure of the GTF file, where all the structural and functional information are annotated. So, if you want to create your own GTF, it should follow this structure, as we said in the first tutorial. If want some examples, you can find them in this subtab. To finish this tutorial, I will demonstrate how to access analysis-specific visualization options. For this project, I have run one analysis. We want to remind you that all data has been created randomly for this tutorial. For now, I will only show some of the plots that are available. First, click on the Differential Analysis button, and select the differential isoform usage results. Next, click on the Data Visualization icon on the left-hand side panel to see analysis-specific results summary plots. We will see all of this more in depth in the next video tutorials. Thank you for watching, and see you in the next video.